For my final local expert project, I have chosen to focus on the therapeutic strategies of three-dimensional stem cell spheroids and organoids. This method can be utilized for many different applications, such as tissue repair and regeneration. So this technique has an extremely long name, but what is it exactly? I'll be explaining this technique by breaking down the technique and its name. We'll start with the three-dimensional stem cells. Three-dimensional stem cells are stem cells that are grown in carriers with 3D structures of different materials and usually various types of cells in vitro. With a 3D structure, cells can actually migrate and interact with each other from all directions to better mimic the function of living tissues and organs. The next word in the technique, three-dimensional stem cell spheroids and organoids, is spheroids. Spheroids are clusters of a broad range of adhesive cell types which create high cell density. Stem cell spheroids are used for studying tissue engineering and regenerative medicine, or more specifically, self-renewal, generation of differentiated progeny, and protein secretion. I will be touching more on the figure on screen later on in this presentation. The final word in the technique's name is organoids. Organoids are tissue-engineered, cell-based in vitro models that mimic the aspects of the complex structure and function of in vivo tissue to resemble an organ. This allows us to mimic organs for experiments to develop future treatments, therapeutics, and even more. Taken all together, the goal of three-dimensional stem cell spheroids and organoids is to better mimic in vivo conditions by replicating cell-cell interactions or cell ECM interactions to develop novel therapeutics and drugs and to observe tissue repair and replacement. I'll now go over the steps that are involved in this technique. First, single cells are drawn closer to form loosely adhesive cell spheroids because ECM fibers and complementary binding at the peripheral cell surface to integrins encourage preliminary aggregation. Next, cadherin on the cell membrane surface induces tight connections between the aggregated cells because of the homophilic cadherin binding of peripheral cells. Following this, early cell assemblies formed by both pathways generate contractile forces via rearrangement of actin stress fibers, leading to compression and the formation of mature spheroids. After this, strong, compact multicellular spheroids are formed. Finally, stem cell-based organoids can be formed by providing the appropriate physical and biochemical signal for differentiation and development of organ-like phenotype, or embryoid body after deformation of stem cell-based aggregates and spheroids. There are two different main 3D cell culture systems that we can use in this technique. We can use a scaffold-based system, which includes using a hydrogel matrix, well plate, or microfluidic chip. With a scaffold-based system, we are essentially providing a handicap or head start to the cells to help them aggregate. More specifically, with well plates, we use low cell attachment plates with surfaces that have modified hydrophobicity to promote strong and fast aggregation of cells. With hydrogel matrix, which is a complex network of physically and chemically cross-linked polymer molecules that expand in aqueous media, we can provide the cells with a head start to aggregate. And with microfluidic chips guiding cells into grooves and holes, we can help cells aggregate this way as well. The alternative to a scaffold-based system is a scaffold-free system, which involves methods such as hanging drops, rotation methods, and magnetic forces. Scaffold-free systems don't involve any biomaterials or ECM matrix, cell adhesions, and surfaces for the cells to grow on. More specifically, with hanging drop, we use cells aggregating at the tip of a droplet of a liquid using surface tension. With rotation methods, we use cells in a suspension in a flask with rotation and stirring that prevents cells from adhering onto a surface. And with magnetic force, we use magnet-labeled stem cells to help these stem cells aggregate to each other. So how does this technique tie into one of our most recent modules? Module 5 was all about culturing and manipulating eukaryotic cells. In this case, we are culturing 3D stem cells as part of spheroids or organoids to use them for later research. In addition to this, we are also manipulating eukaryotic cells through stem cell differentiation. Despite how useful this technique is, there are still several limitations. The first limitation of this technique is that we currently only have developing mature organoid systems and not fully mature organoid systems. This makes it more difficult to study or research anything having to do with organs more accurately, as we do not have access to fully grown or mature organoids. Additionally, if we had fully mature organoids, we would be able to replace organs and regenerate tissue. Currently, we also don't have enough guidelines on the usage of stem cells in a research facility. Thus, we need more guidelines on the appropriate amount of stem cells to use due to how diverse disease mechanisms, biological properties of damaged human tissues, and areas can be. Three-dimensional stem cell spheroids and organoids is also an expensive and time-consuming technique. Finally, we still need better technique to deliver a large number of stem cells while maximizing survival and minimizing cell loss. With limitations also come advantages of the technique, especially when compared to other techniques. First, I want to discuss regular stem cell therapy and how 3D stem cell spheroids and organoids is better than regular stem cell therapy. Regular stem cell therapy, while groundbreaking when first discovered, has many limitations. First of all, regular stem cell therapy has lower transplant efficiency, and if we cannot transplant our stem cells, then we cannot mimic any sort of organs or cell system. Secondly, the stem cells used in regular stem cell therapy have very limited survival rates, while the cells used in 3D stem cell spheroids and organoids have a much longer lifespan. This makes it easier to work with 3D stem cell spheroids and organoids. Going hand in hand with the limited survival rate of stem cells in regular stem cell therapy is the rapid loss of transplanted stem cells in host tissues, making it once again impossible to efficiently and successfully conduct research with these cells. Finally, spheroids and organoids allow for efficient tissue regeneration and repair in vivo, which allows for us to better study using stem cells for tissue regeneration and repair, which is one of the main goals of using stem cells in research. Next, we'll focus on 2D stem cell cultures and the benefits that 3D stem cell spheroids and organoids have over 2D stem cell cultures. 2D stem cell cultures were used in the past and are used now to develop and culture stem cells for research and therapeutic purposes. However, 2D stem cell cultures do not involve 3D structures of different types of cells in vitro. Thus, 3D stem cell cultures are an improvement from 2D stem cell cultures because 3D stem cell cultures better mimic the function of living tissues and organs, and as a result, it narrows the gap between cell-based methods and animal models when studying therapeutics, treatments, and such. Biomaterial, genetic modification, external stimuli, and ECM were and still are methods that we use to manipulate cells to have specific properties that we use to study therapeutics, cell interactions, and such in the lab. However, 3D stem cell culture has proven to be a method that consists of advantages when compared to the older and more traditional methods. Due to its 3D structure, again, 3D stem cell culture is better able to mimic the function of living tissues and organs. As a result, this means that 3D stem cell cultures are also better able to mimic cell interactions, whether it be with other cells or the ECM. In addition to this, 3D stem cell culture can better narrow the gap between cell-based methods and animal models than any of the more traditional methods can due to its 3D nature. The final technique that I would like to compare is 3D stem cell spheroids and organoids to tissue engineering. While tissue engineering can provide the same benefits as 3D stem cell spheroids and organoids, and that tissue engineering can mimic tissues and its environment for research purposes, if the tissues or organs are heavily damaged and we are seeking for solutions on how to repair tissues or organs, then tissue engineering is not as ideal as stem cells can maximize therapeutic efforts better than tissue engineering can. Now that we have spoken about how 3D stem cell spheroids and organoids are better than other techniques that we have for the same purpose, we can now talk about controls. There are really no controls for cell cultures since we are not really expecting any data or lab results, but rather we are just expecting healthy cells that we can work on for research. However, perhaps we can compare the 3D stem cell spheroids and organoids that we culture to previous cell spheroids and organoids to make sure that cell morphology and characteristics are correct. Finally, we can talk about different figures that will further teach us about 3D stem cell spheroids and organoids. This figure shows the change in the morphology of 3D stem cell spheroids cultured in G9 and G6 medium suspension. The G9 medium suspension contains 20 nanograms per mil of epidermal growth factor, or EGF, and 20 nanograms per mil of basic fibroblast growth factor, or BFGF, while the G6 medium suspension contains 10 nanograms per mil of EGF and 10 nanograms per mil of BFGF. In the figure on the slide, we see that spheroids begin to form on day 3 through their smaller and more transparent morphology versus days 7 to 10 when the spheroids are stabilized as seen through their larger and more filled morphology. Taken all together, this figure essentially just shows us what we should expect with regards to morphology of cultured 3D stem cell spheroids when they're healthy. This will help us know whether or not we are on the right path with our own 3D stem cell spheroid cultures. This figure shows us the viability and histology of 3D stem cell spheroids so that we can identify healthy 3D stem cell spheroids through histology. When the 3D stem cell spheroids are stained with acridine orange and ethidium bromide, as in the right side of image A, we see that the viable cells appear bright green and spherical, especially when compared to the stained image of the monolayer cells on the left side. When 3D sections of the 3D stem cell spheroids are stained with hematoxylin and eosin, as the right side of image B shows, healthy viable 3D stem cell spheroids will have red stained images with no showing of a necrotic core. The right side of image C shows us what dot plot images representing stained healthy 3D spheroid cells look like when analyzed by flow cytometry, so that should we choose to verify that we have healthy 3D spheroid cells through flow cytometry, then we know what healthy 3D spheroid cells look like and can verify that our samples are healthy. The final image, image D, just shows us that staining our cell population in the case of both monolayer and spheroid cells doesn't kill any of the healthy cells. The final figure shows us the process of the formation of spheroids in the microfluidic template, what it looks like in real life, and the particle size distribution of the spheroids in the microwells. Image A shows us the process of the formation of spheroids in the microfluidic template in a cartoon. The first step in this process is to load the cells into the microfluidic template. Once the cells have been loaded, the cells will move into the grooves in the template to seed themselves. Finally, with the cells seated in the grooves, the cells will aggregate to form the spheroids. Images B, C, and D show us the image of the bottom layer, the top layer, and the cross-section of the microfluidic template during spherical formation respectively. Image E shows us what the microfluidic template for spherical formation looks like when looked at as a whole, while image F shows us what the spherical formation in microwells looks like under light microscopy. The final image, image G, shows us the particle size distribution of the spheroids in the microwells. In other words, this bar graph shows us the distribution of each differently sized spheroid in the spheroid clusters. Looking at figure G, we see a dumbbell curve, with the most frequent diameter size being 154 micromillimeters. Taken all together, this figure walks us through the process of the formation of spheroids so that we can know what to expect in a proper spheroid formation. This can help guide us so that we know whether or not we are carrying this out correctly based on the morphology of the spheroids that we are seeing in our own cultures. Thank you for listening to my presentation. I hope that you enjoyed it. I will be showing my sources on the next slide. If you want to stop and look at some of the sources, please feel free to pause the video. Thank you again.